The talk is called Science Discovers Intelligent Design in the Foundation of Life. And it tells the story of science, the results of science, displaying the intelligence of living things and how we recognize that. So let me say that some of you know, over the past 25 years, I've been interested in the question of intelligent design and Darwinian evolution, and I've written several books about it. The first, Darwin's Black Box, which I talk about the biochemical machinery of the cell and why that points to purposeful design. But just because there's some design in life doesn't mean that everything had to be designed. So a while later, I wrote the book called The Edge of Evolution, which asks the question, how much can unintelligent processes in nature do, and how much required design in life? And, the third book recently came out, talks about the surprising result that Darwin's mechanism is powerfully devolutionary. It works mostly by breaking things. We'll probably talk a little bit about these in the second half of the talk, but the point I want to make is that these three books all make the case that Darwin's mechanism is completely inadequate to explain the molecular structure of life. But believe it or not, that's actually not as controversial as the second point. The second point in the books is that purposeful, intelligent design is required. The first point, everybody admits, or many, many people admit. For example, if you go on the internet, you can find a website called the Third Way of Evolution. And on the first page, it lists a number of scientists and academics who think that neo-Darwinists have elevated natural selection into a unique created force without a real empirical basis. That is, these prominent people in science and other fields think that the evidence for Darwinian evolution is really inadequate. So because of that, I'm going to take that as given by most people, that Darwin's mechanism doesn't really do what it claims to do. And I'm going to focus initially on that second point, that is that intelligent design is a better explanation for the foundation of life. So before we do that, I'm just going to show you a few examples, a few interesting examples of what leads us to think that. And the first example is from experiments that were done, maybe about 5 or so years ago. And 5 or so years ago, some English microscopists looked at a common insect called the plant hopper, and they used a new microscopic technique that allowed them to look even more closely at some mysterious structures that were known to be on the insect's legs, but nobody knew what they did. They were just little bumps that people had noticed before. But using their new technique, they were able to show that those bumps were gears, real hard mechanical gears. And this is the first time that such macroscopic gears have ever been seen in living things. And it set off a discussion on the internet, a theological discussion for a couple of weeks about this. But then people kind of went on and started to talk about other things. But they found this disturbing. And the question is, why did they find it disturbing? Another example that I'd like to show you before we start talking about design is the good old bacterial flagellum. I expect that most people listening to this talk are familiar with the flagellum, which is an outboard motor that bacteria use to swim. It's got a propeller that spins around and around, and it's got this region to hold it onto the drive shaft, and it has a motor and many more parts. And I actually wrote about this, in the mid-90s, and argued that it was beyond Darwinian explanation, that it was irreducibly complex, and therefore could not be explained by Darwin's mechanism. And at the time, in the 1990s, this set off a firestorm of criticism, that people said that was ridiculous. Many scientists said that, oh, Darwin's theory will certainly explain it soon if we haven't done so already. But in my book published last year, I show that decades after that uproar, decades after the challenge to Darwin's mechanism, that no Darwinian biologist or anybody else has even tried to explain in real terms how such a structure could have come about. Instead, they essentially just turned to talk to other things. So the challenge still stands, even though very, very smart people were infuriated by the claim of intelligent design and were very motivated to show that it was wrong. It still stands as a mystery to Darwinian theory. But progress has been made in the past 25 years, but not progress in explaining it by Darwinian terms. Instead, progress in understanding how the flagellum works. I want to concentrate here on this structure. This is called the hook region, and it acts as a universal joint. A universal joint can change the plane of rotation of one element, the drive shaft, into a different plane of rotation, the propeller. As the drive shaft turns, this is a mechanical part that turns around and around and around and around and around. If that motion were transmitted here, then this whole thing would turn around and around and around and around. If these were hard parts that transmitted this force. But instead, it's seen that this motion around here is transmitted so that this spins just like this, in this plane, not in this plane. So this doesn't go woo, it goes in just this plane. And some scientists a couple years ago showed that used a new technique to look at just this hook region. Used a technique called cryo-electron microscopy. And one thing you have to realize is that the parts of the flagellum, what was shown on the last slide, is just a little cartoon, and that each of the parts is made up of many complex protein molecules. And I think you can get a better understanding of that by looking at this detailed part in here. This is composed of a number of proteins, and each one of the little dots is an atom in the protein. And the workers showed just last year, 2019, that the hook region acts, this is going in this plane, but this is going in a different plane. And this works by the center staying together, but the outer parts of the proteins that make up the hook region, they come closer together on the inside and further apart on the outside. Closer together, further apart, closer together, and further apart. And that allows the structure to act as the universal joint. It takes a large number of atoms in the right positions, very many interacting parts. And that is just one element of the bacterial flagellum. Okay, I want to show you another structure that has been looked at in recent years by this new technique of cryo-electron microscopy. And that is this thing. It's called a bacteriophage. It looks like a lunar lander or some exotic structure. Bacteriophages are viruses that infect bacterial cells. And when they get inside, they essentially eat the cell and destroy it. Here's an electron micrograph, an old technique, the electron micrograph at room temperature, showing it attacking a bacterial cell. And this is kind of an artist's drawing of what this looks like. But again, recently, some people looked at just a portion of the bacteriophage. They looked closer into what's called the tail region and the base plate. And they showed that, in fact, the structure is very, very detailed and complicated. You see this rod, which in the complex before it attaches to a bacterial cell is extended, but when it touches a cell, it compresses, and a needle-like structure comes out of here, penetrates the cell, and injects DNA into the cell. And again, let's look at this area here. The workers showed that when it touches, when this touches a cell, these tail fibers extend and grip the cell. Now I show you this because I want to show you the precise mechanical properties of the structure, that even this is a cartoon, and that many, many, many different interactions go into making the structure actually work. So the descriptions of things that I gave in Darwin's Black Box were very superficial, and that the problem for Darwin's theory is much, much, much worse than I had said. Okay, so when we see things like that, we are strongly inclined to think that those things look like they were purposely designed. That didn't occur by chance. A question I want to discuss now is, why do we think that? Why do we think that those machines, those cellular machines, were indeed designed? Well, the person who is credited with first discussing it is a man named Thomas Reid, who lived in the 1700s. And Thomas Reid was a Scottish philosopher, and he was interested in questions of the human mind and how we think. And he wrote about something he called the design principle. And that, he said, is the general principle that intelligence in the cause can be inferred from its marks or signs in the effect. That is, we cannot see intelligence. It's not that we look at a person and see their head glowing when they're thinking. The only way we can tell that there is an intelligence is through its marks or its signs. Well, how do we do that? What are the marks or the signs of intelligence in the effect? Well, if you look at a dictionary, a lot of dictionaries will have a definition of design, and one common one is seen on the internet. The design is the purposeful arrangement of parts or details. And we can simplify that just to say that design is the purposeful arrangement of parts. And so that means that we recognize design, we recognize a mind has worked whenever we see parts that seem to have been arranged for a purpose. Well, let's, instead of just resting with that definition, let's look to see an example of that, of parts that appear to have been arranged for a purpose. This is a cartoon that I like to show audiences. It's a cartoon called The Far Side, which is very funny. I like it a lot. And it shows a troop of explorers here. And the lead explorer has been strung up and skewered. And this fellow here turns to this guy here and says, "That's why I never walk in front." Words to live by. Now, everybody viewing this looks at the cartoon and you immediately know that this was designed. You know, it's not an accident. It's his death was intended. As a matter of fact, the humor of the cartoon depends upon you recognizing the design. You know, if he had just fallen over a cliff, it wouldn't be quite as funny. But how do we know that? How do we know that this was purposely arranged? Well, we know that it's designed because we see a number of parts that have been arranged for a purpose. So all these parts work together for the purpose of killing this poor jungle explorer. So this is how we recognize that that a mind exists. It's by external things, only by the actions that a mind takes. Here's another example of the same thing. About 20 years or so ago, a man named Jean-Dominique Bauby, a French man who was editor of a French magazine called Elle, suffered a stroke. He was driving and all of a sudden his head began to hurt and he suffered a stroke. And as a result of the stroke, he couldn't move anything. He couldn't move at all. He was paralyzed, except that he could blink his left eye, his left eyelid. That's the only motion that he could undertake. If somebody walked through the hospital, they would just see a person laying in bed, unable to move, and they might not think that he was conscious at all. But it turns out that he could blink his left eye, and he blinked his left eye to a translator. And he blinked it in code, and the translator wrote down what he was thinking, or what he signaled he was thinking. And he wrote a book He wrote an entire book called The Diving Bell and the Butterfly, describing his experiences in the hospital and what it was like to be paralyzed. So the point is that looking at him, you wouldn't know if he had a mind or not. It's only by the purposeful blinking of his eye, the purposeful arrangement of parts, that we can tell that there is that it is the work of a mind. It turns out that the purposeful arrangement of parts is the way, the only way to recognize the existence of a mind. As you know, this is a famous sculpture by the French sculptor Rodin called The Thinker. And we are invited to suppose that as he's sitting there, something is going on in his mind. But we can't tell. We can't tell until he acts and he begins to arrange things in for a purpose. Okay, so the purposeful arrangement of parts. But there are a couple of other points I need to make, or else we could become confused about this. And one of the points is that the force of the argument is quantitative. That means that the more parts that are arranged and the more closely they fit their purpose, then the greater and greater is our conclusion that the work was designed. And let's take an example of this. These are the Sawtooth Mountains in Idaho. And if you look at them, you say, well, they look just like mountains. There's no reason to think they were purposely designed. But if you look at another structure of another mountain, this is called the Old Man of the Mountain in New Hampshire. And this structure looks like the side of a face. It looks like here's a chin and maybe a mouth and a nose and a forehead. So you might say, well, did some prehistoric tribe, put all of these boulders in position? And you say, no, it's just an accident. It's kind of cute. It looks like a face, but it's not really. It's probably just an accident. But if you look at another mountain over here, this is Mount Rushmore in the United States, with the faces of 4 past presidents of the United States. Anybody looking at this would know that these figures were purposely designed. And the reason that you would know it is because you see that the parts of the mountain, the shapes of the rocks, are purposely arranged in order to present these faces. So the point is that the conclusion of design is quantitative. You can have no evidence for design. You can have a little bit of evidence for design, or you can have overwhelming evidence for the purposeful arrangement of parts. Okay, and there's one more point that I have to make, again, to make sure we don't get confused. And that is that design can occur on multiple independent levels. Okay, there doesn't have to be just one structure completely designed. You could have multiple levels of design. And here's an example of that. If you go into a lumber store, you can see little blocks or pieces of wood or boards and so on. And if you looked at those, you'd say, these appear to be purposely designed because their parts have been arranged so that they can act as building blocks of other structures. If you looked at a building that was being built, when you saw these things arranged so that they're forming a roof, you say, well, this is not an accident. There wasn't a child just nailing boards together. Somebody who knew what he was doing put these boards in this arrangement. And when you see the finished product, these boards could have been arranged into any kind of a house or lots of different structures. But in fact, they have been arranged into this specific structure. And you can say this in fact, was designed too. So you can have different levels of design. And that's kind of important because there's a debate among different factions of people who say they think intelligent design is empirically discoverable, but they differ on the degree to which it is discoverable. For example, it can be likened to saying that Mount Rushmore was designed, or that Mount Everest was designed, or Mount Rushmore was designed. Here we have two mountains, but there are some design proponents who say that if we look at the laws of nature, we can tell that they were designed because they allowed the universe to unfold with great specificity to give us what's called a Goldilocks universe. That is, it's not too hot, it's not too cold, it's just right for life. And we can tell that the universe was purposely arranged because the laws are fine-tuned for living things. And that's fine, but many of them then say, we have to stop there. We have to stop there. We have to think that the universe was designed, but that life and the unfolding of life over time was not necessarily designed. That's a different level. We know that the foundation level, the physics, was designed, but we think that the level of life was not necessarily designed. And I disagree. I think that we can tell that the physics level of the universe was designed, and life too required more design, just like Mount Everest. You know, the existence of mountains and so on required the laws of the universe to be set correctly. But they don't explain, the fundamental laws of the universe do not explain the faces on Mount Everest. And we can kind of make an analogy to a kaleidoscope. A kaleidoscope, as you know, is a toy which has little colored pieces in one end. And as you turn it, it makes the pieces tumble. And if you look through the end of it, they can make all pretty patterns because there are mirrors, symmetrical mirrors lined up so that it reflects on itself. And you can say, wow, the inventor of the kaleidoscope purposely arranged the parts so that it could form these pretty patterns. And here's another very pretty pattern. And you can liken those, the structure of the kaleidoscope, to the laws of the universe. But if you turned the barrel of the kaleidoscope and you got an image like this, you would say to yourself, wait a minute. There is some reflections here and pretty pattern, but these things, this looks like eyes and this looked like a nose and stuff. This is making me think that there is another level of design beyond just the structure of the kaleidoscope. And if you see a structure like this, you'd say, forget it. This image of a cat is not explained just by the structure of the kaleidoscope. It required purposeful design beyond just the kaleidoscope, beyond just the laws of nature. So again, we can say that, yeah, it's true, the universe required design of its fundamental laws, but it required more design to produce the structures like the bacterial flagellum or the plant hopper leg gears. Okay, and this is actually I don't know if you can see it. It's called Kaleidoscope Cat, and it was drawn by a French artist. So we know that it was purposely designed. Okay, so in the last 15 minutes or so of my talk, now that we know how to detect design, I want to just quickly reprise the arguments that say Darwin's mechanism, which many very smart scientists say can mimic design, can look like design, can't do so. The problems with Darwin's theory. And I started this in the mid-1990s with Darwin's black box. And the main point is that Darwin proposed a criterion that would show his theory to be false. And he said that if we could find anything in life that couldn't be produced by numerous successive slight modifications, then his theory wouldn't work. And in the book, I said, well, what sort of a system could not be produced that way? And said that something like a mousetrap, something that was what I called irreducibly complex. And that just meant that it needed a number of interacting parts that would have to work with each other to produce some function. Just like a mousetrap needs multiple parts, a spring and a hammer and platform and so on. You need all those parts in order for it to work. And you cannot produce something like that gradually by small steps, because it pretty much doesn't work until the whole structure is assembled. And then I went on to say that there are many things like that at the molecular level of life, the foundational level of life, such as the bacterial flagellum that we talked about. And I didn't even mention at that time, it was not even known how the components of the flagellum interacted as they did. But I want to point out here that irreducible complexity is what is called a twofer. That means you get two different features in one structure. Irreducible complexity is, first of all, an obstacle to Darwinism, because irreducibly complex systems can't be produced by Darwin's numerous successive slight modifications. But they're also powerful evidence of purposeful design, because you see a number of different pieces arranged for a purpose. A number of pieces arranged for a purpose in a flagellum. Okay, that was the argument, and the next couple of books that I wrote wanted to test the edge of evolution. Sure, Darwin's mechanism can't make a flagellum, but can it make other things? And I'm going to go to the more recent book, Darwin Devolves, because it has more surprising results. And in it, I show that, or I argue that science advances a lot when it has new technologies, new instruments that it can use. For example, when Galileo used the telescope, he discovered that the universe was a lot bigger and a lot more complex than had been thought to that time. When the microscope was first invented, we could realize that life existed at a level, a microscopic level, and smaller than anybody had ever suspected before. We didn't even know that such things existed. Well, we're living through such a time right now, a time where new technology is allowing great advances in science. And in particular, we have a revolution in our ability to sequence DNA. As you know, DNA carries the genetic information of life, and it does so in the exact sequence of the parts of the DNA called nucleotides. It's only been in the recent couple decades that technology has been developed that allows us to determine the exact sequence of DNA and to do it in sufficient detail and with enough organisms to ask the question, how is DNA changing as a species evolves? Before this, scientists could say, well, look, this organism was born and it has a different feature. Maybe its fur is white and its parents' fur was brown. And so we could say that it was evolving, but we couldn't say why. We didn't know what the changes were at the DNA level that gave it this new trait. And in order to properly evaluate Darwin's theory, we have to look at the molecular level of life, at the level of DNA, because mutations are changes in molecules. And changes in DNA lead to changes in proteins. So we have to look at that level to properly evaluate Darwin's theory. And this man, named Richard Lenski, has been doing this for the past 30 years or so in bacteria. He watched bacteria evolve in his laboratory over 60,000 generations. And because of the development of technology, he was able to sequence the entire genome of the bacteria, E. coli, and he could see when a bacteria had a mutation that gave it an improvement, allowed it to reproduce faster and outcompete other non-mutant bacteria. He was able to track it down and see what those mutations were. And in an early paper in the year 2006, he looked at a couple of genes. Each of these bars is supposed to represent a gene of E. coli. And he saw that mutations in many places in these genes led to improvements. That is, the bacteria could grow faster. And he noticed that that signature was the signature of mutations that are helping by breaking or degrading the gene. And that's because a gene can be broken. A gene is a complex structure, and it can be broken in any of a large number of places. Just like your computer can be hit with a hammer at a number of places and it would break. But if you wanted to improve the gene, there's only generally one or two at most places where you could change it and have it be better. So it turned out that those 4 genes were helping by being mutated so that they were broken. And then in the year 2016, he showed the results of 50,000 generations of evolution of E. coli in his lab. And he showed in a couple of tables that all of the genes that helped, all of the beneficial changes, were ones that broke or degraded the genes. And so none of them actually were building new structures like the machinery that we saw earlier in this talk. They were all degrading structures. But you might ask yourself, how can breaking a gene or degrading something help? Well, here's an analogy on the next slide. Helpful degradation. Suppose you own this nice car. This is a very nice car. But suppose that your life depended on you getting better gas mileage. If you could go a little bit further on a tank of gas, you would live. But if you didn't, you would die. Well, in that case, then getting rid of excess weight, getting rid of the hood or the doors or the back seats, that would lighten the weight of the car and allow you to get better gas mileage. Of course, doors and hoods of trunks and so on, they're useful in many circumstances. But if the conditions were such that your life depended on getting better gas mileage, then it's best to throw things out. So it turns out that this is the way that evolution proceeds in the great majority of cases. It's not just bacteria. Scientists have sequenced the DNA of many different dog breeds too, precisely because they're interested in determining how such a variety of dogs has been produced over the past few hundred years. I don't have time to go into it, but they tracked down the different breeds, the features of different breeds to different genes. And most of them, the great majority of them, had suffered mutations that degraded the gene or outright broke it. Many other species too, including a polar bear, the entire genome of the grizzly bear and a polar bear have been sequenced in recent years. And precisely, again, because scientists wanted to see the genes that had changed to make this new species. And they found that the large majority of them, actually more than 50%, were ones that degraded the genes. So the point is then that evolution, Darwin's mechanism, does work, and it has important consequences. But it works mainly by degrading or destroying genetic information that was already there. It doesn't put new information into the genome. Okay, so what can Darwin's mechanism do in terms of the evolution of organisms? Well, a good place to look is at the examples Darwinists and evolutionists point to of species that have evolved quite a bit over in the recent past. For example, an icon of evolution are the different species of finches on the Galapagos Island. And scientists point to those as an example of the power of Darwin's mechanism. Look at all those species that have been developed. And it's thought that they've developed in the past 2 million years, not just in the recent past, but in 2 million years. Okay, well, it's interesting, but if you look at the 8 major levels of biological classification, domain, kingdom, phylum, class, order, and so on, well, all of the species of finches, they're all eukaryotes. They all share the same domain, and they're all animals. They share the same level at kingdom, they're all chordates, animals with backbones. They're all birds. They're all passeriforms. They're songbirds, and they're all in the family Thropidae, all of those species of finches. They only differ at the level of genus and species, the two lowest levels of biological classification. Now, each of these levels is very different from each other. And we can liken these, say, to a sum of money that's in the hundreds of thousands of dollars, including cents. And each place value in the number is a different biological level. So let's look at the, oh, I'm sorry. So here is the line. In 2 million years, none of the finches has been able to evolve past the level of genus. And if we consider each of those levels to be a number in a sum of money, including hundreds of thousands of dollars and including the cents position, then all of that evolution occurred in the very lowest two levels. It occurred only in the cents, the pennies level of this huge figure. So it's a very, very small amount of evolution. Another example are things called cichlid fishes, which live in what are called the Great Lakes of Africa, Lake Tanganyika, Lake Malawi, and Lake Victoria. And if you look at different species from different lakes, they're different species, but they oftentimes resemble each other very closely, even though they've been separated from each other for millions of years. And if you look at the new species that have evolved in the Great Lakes, it's the same story. They are all eukaryotes, they're all animals, and so on. It's only at the level below family that you get new species or new genuses. So there seems to be a line at the level of species, or level of genus, or below the level of family, where you cannot evolve new forms beyond that. Okay, so those were finches and cichlids, but there's a bunch of other organisms, kinds of organisms that evolutionists have studied that have undergone lots and lots of, have produced lots and lots of new species. Anoles, birds called honey creepers, fruit flies, plants called silver swords and lobelias. And yet, none of them were able to produce an organism that differed from the ancestor at the level of family. They were all confined to species and genus. So it's a reasonable thing to conclude that Darwin's mechanism, yes, it can work, and it can work at lower levels of biology, the level of species and genus, but it can't make anything that differs from the ancestor at the level of family or higher. And what is family? Well, in the mammalian order of Carnivora, it's dogs versus cats versus bears and so on. So starting with a generic mammal, you can't get a dog from a cat, or you can't get a cat from a dog. These required separate designs. Okay, that's pretty much my talk. I've gone over time a little bit. I'm sorry about that, but I just want to end by again showing you the greater and greater and greater elegance and complexity of life that's being discovered as science looks deeper and deeper into life. Here's a photograph of some cells in our retina that act as fiber optic cables connecting the outside of the retina to the photosensitive cells on the inside. In other words, these cells are built to channel photons exactly to the receptors where they are needed. And this was a huge surprise when it was first discovered. This had been an example of something that no engineer would have done, to have put the photosensitive cells on the inside. But now we discover that the light is channeled by mechanisms from the outside to the inside of the retina. Another example I just want to mention briefly is something called a magnetosome. A magnetosome, and this is a structure that allows bacteria to sense the magnetic field of the Earth. And these are particles of the magnetosome lined up as they're supposed to be in order to do their job. And there are a dozen different proteins, many different structures that are required just to make this. And if just one of the proteins doesn't work, then you get the pieces of the magnetosome scattered throughout the bacteria, and it can no longer work. It can no longer function. Okay. We don't have time to go into the many details that are necessary for this to work, but I think this picture gives you a little idea of it. So again, we recognize design by the purposeful arrangement of parts. And as we probe life deeper and deeper, we see more and more purpose in more and more parts. And so we can be very confident that a conclusion of intelligent design is rationally justified. Now, not everybody agrees with me on this. I know it seems surprising. And I do have some critics. And if you want to look up my responses to them, they're at several different places on the internet. You can look at my faculty website at Lehigh University, and you look at the website of the Discovery Institute. And finally, I have my own website these days, and I have many responses to critics there. So if anybody wants to get into the real nitty-gritty of this, then these are the places to go. But thank you for your attention.